Hello, in this video we will discuss about the mismatch repair MMR, DNA repair and DNA mechanism we will discuss, and methyl directed mismatch repair we will understand, and mismatch basically is the nucleotide excision repair as well as the mismatch repair. So the nucleotide excision and mismatch repair we will discuss in this video, and mismatch repair and repair the nucleotide excision. So this is the basically is the cell which that contain nucleus, and nucleus contain chromosome, while the cytoplasm, nucleus and the cell membrane. So remember, this is a chromosome. Chromosome contain a chromatin material which that is made up of histone protein and the DNA, 40% DNA, which that here is the adenine will also bind with the thymine, and the basically governing with the cytosine through hydrogen bonding, anti-parallel strand phosphodiester bond containing backbone of the DNA, which that is anti-parallel, mean 5 prime to 3 prime and 3 prime to 5 prime. So this is the UV radiation which that will damage the cell. So in this way, as you can understand, this is the DNA. After the nucleotide excision repair mechanism we will discuss. So in this way, the pyrimidine dimerization or dimer formation. In this way, the cytosine with thymine will connect together is known as a nucleotide excision repair due to this pyrimidine dimer formation. So in this way, many microbes in the environment commonly encounter ultraviolet light, which can cause neighboring pyrimidine, base, thymine or cytosine to form dimers, and thymine dimers are the most common. So remember, so many microbes in the environment commonly encounter the ultraviolet light will affect to cause a pyrimidine dimer formation, and the nucleotide excision repair we will discuss how it is possible. Let's begin to understand. So in this way, here is the 5 prime to 3 prime and 3 prime to 5 prime anti-parallel strand containing adenine, thymine, guanine and cytosine. So in this way, here is the activation of the A subunit containing protein, the exonuclease enzyme containing the 3 subunit. These 3 subunit 2A and 1B. In this way, also the UVRC and UVRB basically and UVRA, which that is the protein. So the this is activity for the exonuclease. So these three type of protein which that is combined together, but the three subunit is the trimeric formation through UVRA and UVRB by the using of ATP to bind the site of pyrimidine dimer formation, and more ATP phosphorylation, the hydrolysis of the ATP will lead to combine the B and A UVR, and in this way this is involved the 12 to 30 nucleotides involved, and the basically further ATP use hydrolysis of the ATP will lead to more UVR, UVRA and B with combined with the number of 1 is the ATP use, and in this way the releasing of the A subunit both, while the C will bind. The recruitment of the C is the second step. In this way, the binding of the UVRC and the B combined together will recruit the UVRD helicase enzyme. The UVRD, which that basically create the nick, and this nick creation will lead to basically the also the preparation by the helicase enzyme which that will lead to helical structure, helicase enzyme, which that lead to unwind. After this unwinding, here is the basically is the removing of the helicase will remove the damaged or the dimer formed cytosine with thymine will remove containing the sequence, which that sequence is 12 to 30 nucleotide involves. So remember, this is a helicase enzyme which that will lead to remove by the creation of nick. After this, here you can see 5 prime to 3 prime direction and 5 prime to 3 prime direction. That will be hydroxyl group will be create. This hydroxyl group, which that will synthesize from 3 prime to 5 prime direction through a DNA polymerase. So here is the DNA polymerase which that resynthesize of the DNA from 3 prime to 5 prime site. While on the other hand, the DNA ligase enzyme, this DNA ligase enzyme used to ligation after the completion of the polymerization, so the gap will be clear by the DNA ligase. So the completion of the dimer formation, which that will lead to cytosine guanine, which that will be separate. So anyhow, let's begin to understand the mismatch repair. That was the nucleotide excision repair, and now we will discuss about the mismatch repair. In this way, here is the DNA which that contain a methylated segment, the guanine, adenine, thymine, and cytosine. But here you can see the rounded line. Basically, the guanine and thymine is the mismatch, and in this way it will target to the signaling of the through DNA will lead to mute S and mute L recruitment and ATP. So this is the methylated sequence involved. So in this way, here you can see the mute H also involved. The mute H basically mute H will bind with the sequence, extra sequence, which that is both side is equal. For example, the palindromic sequence. This is the palindromic sequence which that will bind with the mute H, and this mute H will bind, while here is the mute S and L which that combine where the mismatch is occur, while the palindromic sequence will recruit the mute H which that will bind. After this binding, here the mute S and mute L which that is bind here, and UVRD will recruit, and this recruitment of the UVRD as well as the endonuclease enzyme. So this in this way the UVRD will be separate the strand. After the separation of this strand, the antiparallel strand, so in this way the ATP is used for the endonuclease enzyme to convert the ADP hydrolysis, and in this way the UVRD which that will remain the maintain of the strand, broken the strand. So in this way, here is you can see the DNA polymerase 3 and DNA ligase enzyme used to guanine and cytosine the changes of the mismatch repair. This is the point mutation can be DNA polymerase 3 and DAM methylase enzyme. This DAM methylase enzyme used after the ligase enzyme to the palindromic sequence to again methylate it. So the methylation will again occur through a DAM enzyme. So after this, the methyl group addition means this will be clear the more epigenetic homeostasis, epigenetic maintenance. So this is the mismatch repair. We had discussed about the mismatch repair and nucleotide excision repair in the case of mismatch repair and the DNA repair and DNA mechanism. So I hope you make sense about these things and this lecture. If you like this video, please make sure to subscribe, like and share, and ask the question for answer and comment below.